hi, a very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So thank you so much for joining my session today. Yeah, and please don't forget to like, share and subscribe. So we are here towards the last lecture of biotech. So I wish we say goodbye to biotech here because it's been already 5 lectures. Though we have done it in bits and pieces, so it's only for 20 minutes, 30 minutes lecture. So, okay, so there's someone called as 𝗍𝐴𝐴𝐴𝐴𝐴𝐴𝐴. Hello. So you are asking for 𝐂𝐨𝐴𝐴𝐴𝐴 ma'am. No, 𝐂𝐨𝐴𝐴𝐴𝐴 ma'am is not here with us. So probably you cannot see her lectures in 2022. Okay. Yeah. Anyways, thank you so much for joining my lecture here. So please do like, share and subscribe. Well, so also I would like to tell you, so things are back. I mean to say the pandemic is back. The lockdown is back. So the normalcy of the lockdown is back. So please stay safe and take enough precautions so that you don't get or you don't put yourself in trouble. So make sure even the slightest of infection is dangerous. So please avoid everything. So avoid going to a place where there is a crowd, where it's a gathering. So avoid crowds. That is what I would say. So I know marriages, parties are tempting. I would always go for a change of mind, for looking forward to friends and all. But please, at this time, I wish and I pray that you all stay safe, right? So yeah, so in different parts of India, so various actions are being taken. So prevention measures are taken to stay away from a gathering, to stop the spread of infection. So please stay safe. And I also would like to tell you that you guys have to take the necessary precaution to educate those around you. So make them also feel safe. So don't panic if someone shows symptoms. So just quarantine them, stay safe, eat healthy food. So your lifestyle is what is going to bring you a change, and your lifestyle is what is going to give you immunity, right? So make sure you boost up your immunity. So please don't be careless for all those who are vaccinated. So it does not mean that those who are vaccinated will not get any symptoms. So please stay safe. At the end, this is the information that I want to pass on. Yes, ma'am, a very happy good evening. So thank you so much for joining me this evening. So please do like, share and subscribe. Well, so talking about biotechnology part 6. So now comes the next question. Ma'am, we are having lockdown. So will we have term 2 board exam? When will we have NEET exam? So guys, we cannot foresee the future, so I cannot tell you anything about the future. So future is so uncertain, right? So as of now, the classes have paused. They have told us to ease the offline classes. They have all told us to take us preventive measures and to stay safe. So what if the spread of infection declines by the start of March, and in March everything comes back to the new normal, and they say you have term 2 exam and the NEET exam is in the month of May. So that time you don't have much amount of quality time to do or study or start a chapter from start. So I would say, even though the lockdown is here, so though you are supposed to stay at home, so please follow your original discipline. So please religiously follow the timetable that you have sticking to. So never take a break. So don't be like, yeah, the NEET exam will be postponed because the 3rd wave has come, so let me also take a gap. No. So you can probably increase the number of breaks that you take, but please follow your schedule. So that's what I will say, because as the lockdown extends, as the NEET exam gets postponed, the competition level will also rise, right? So please think from that point of view and start studying. So please don't take time for granted, because time once wasted will never come back. All right, so here we are going to biotechnology. This is part 6. So we are done with 5 parts. So probably this is the last part, so where I'm going to put all the pieces together and explain you. So far we were talking only about the tools of recombinant technology. So now I'm actually going to tell you about how the process of recombinant DNA technology happens, because we haven't spoken about how to isolate DNA or how to isolate plasmid. So that is what we'll be talking now. Hi W, yes, a very happy good evening. So thank you for joining my session this evening. So please do like, share and subscribe. Chalo, let's move forward. Yes, ready? Yeah. So I'll make sure I'll finish it today. I will not extend more. So we were talking about recombinant DNA technology. So probably I was talking only about the tools of recombinant DNA technology. So the tools include your restriction enzyme. So then you had the cloning vector. Then the last one was a competent host. So how to give that heat shock treatment to make sure that your bacteria has taken up the recombinant plasmid. So that is where we ended our session on Saturday. Yes, I am very good. How are you doing, W? Are you doing good too? Yeah. So these were the tools that we were talking about. And for each of the two, we also have a cross verification step to see if that is there or not, right? So to see if your restriction enzyme has worked or not, so we do gel electrophoresis. And from the gel, we isolate the DNA, and that is elution, to check if your plasmid has ligated with your gene of interest and to make sure your plasmid has entered the bacterium. So we do positive and negative selection. So we can probably do antibiotic selection, or we do insertional inactivation. So this is where we stopped, and we were done with part 5, or like 5 lectures of your biotech. So now how are we going to proceed further? So ideally a concept of recombinant DNA technology. So our concept of DNA technology is to take, so if we have a plant A, so we have to isolate the DNA of plant A, right? And then use restriction enzyme so that you get only a gene of interest, right? So side by side you will also isolate a plasmid or construct a plasmid, and you will cut open the plasmid with restriction enzyme, same restriction enzyme, and then you have to ligate your plasmid and your gene of interest together. Now you will have to put it inside a bacteria and make clones. Sometimes you just need to take sexual reproduction in flowering plants. So probably sexual reproduction in flowering plants is done, W. So you can look into this New Beginning series. So you have all my videos of sexual reproduction in flowering plants. So probably I think I have taken 5 lectures of sexual reproduction. So I have recently done last month. So please look into that video. So if you don't understand, I am ready to explain, but please just once go through the lectures. Alright? Yes? Fine. So we produce clones in the bacterium. So once you produce clones, sometimes our end product is to isolate the plasmid alone, to isolate the clones, and then put it inside plant B. So this is our entire process of recombinant DNA technology, right? So now here I have the process listed out like this. So isolation of DNA. So first, if I am taking a DNA, if I need a gene of interest, for example, I am taking the gene of interest from plant A. First of all, I have to isolate the DNA of plant A. So how do I do it? And accordingly, I have to isolate a plasmid if I am taking a plasmid from a bacteria. Right? So how do I isolate? We are going to see. So fragmentation of DNA by restriction enzyme, this step is discussed. So separation and isolation of desired fragment, this is discussed. Gel electrophoresis and elution. Amplification of the GOI using PCR. For example, if I get my gene of interest as a small fragment which is only 100 kilo base pairs, so I need to produce clones of it and keep, because there are high chances I miss it. For example, say I have taken a Dinosaurus gene. So Dinosaurus gene is say for example I have taken it from a fossil, right? So I am isolating the Dinosaurus gene, and from that I am taking one gene of interest. So once again, I am not sure if I'll be able to isolate the DNA or if I'll get access to that fossil of the dinosaurs. So I have to make sure I produce clones, or I amplify it, or I produce many copies of this gene of interest. Now, how do I produce many copies of this gene of interest without putting it inside a plasmid? Is I use PCR. So what is a PCR and how is it done? We will see. And once the gene has been cloned, you insert your gene of interest inside the plasmid. So ligation of DNA fragment into the vector. Now transferring of this recombinant plasmid into the host, right? And probably into the bacteria, so that you produce more copies of your recombinant plasmid. So culturing the host cells in a nutrient medium. Say for example, you're directly putting it inside plant B. So now you have to grow plant B so that the gene of interest is actually expressed, right? So culturing the host cells in a nutrient medium at a large scale, and then you take the extraction of the desired product. For example, if I have to make, or if I have to extract an enzyme from the bacteria, right? So I take the gene of interest, for example, a gene which codes for my enzyme. So I will clone the gene of interest and then I will put it inside a plasmid. Now I need more copies of this plasmid. I put it inside the bacteria, so it gives me more copies. So now my end product is to isolate that gene, or if that gene is expressed inside a bacteria, the bacteria will produce what? The enzyme. And now my process is to only isolate the enzyme. So what I am saying, so this is an example. So suppose if I want to produce an enzyme, say for example, I need lipase enzyme. So lipase enzyme is actually used in your detergents to remove oily stains, okay? So I take the lipase gene of interest. So I do PCR and I make clones. I make it and store it in a library, okay? Yes? So I stored this, so I do PCR, so I produce clones. So I have many copies of my gene of interest. I store it in my library for future reference. So few copies I take and I put it inside a plasmid like this. Fine? Yes? So now this is my recombinant DNA. So now I put this inside a bacteria. So the plasmid gets expressed and this gene of interest produces my lipase enzyme. So lipase enzyme is produced inside my bacteria, but now I will have to isolate and purify this lipase. So this is also my recombinant DNA technology. Alright? Yes? Tech geek. Probably I forgot you. I'm really sorry. I don't know, but I don't know if you have changed your name. Yes, a very, very happy new year. So thank you so much for remembering and getting back to my session. So please don't forget to like, share and subscribe. Wow, thank you so much for your wishes to my students. Yes, definitely. I hope they come out with flying colors in the NEET exam. Thank you, Tech Geek. Right. So this is also a part of recombinant DNA technology. Okay. So the points that are in black color are the ones that I am supposed to explain. So probably I have to explain only 4 points and then the chapter is done. So now let's see how to isolate a plasmid or a DNA, right? So isolation of genetic material, DNA or plasmid, it's Tech Geek. Acha, you're Vishal. So are you Vishal? Okay, so here we go. So the bacteria first, so if I have to isolate the gene of interest from a bacteria itself, so I will make sure I will grow the bacteria well. For example, if you're going to eat chicken, you will make sure you feed the chicken well, right? So much in the same way here, I'm not going to kill the bacteria, but I'll culture the bacteria, I will grow them and then I'll harvest them. The cells are broken to release their contents, because I just need the genetic material. The genetic material is present inside the nucleus, right? So now, how do I break the cell? So for example, if I am talking about a plant cell, so plant cells are very strong in the sense that they have a cell wall. So now I have to break this cell wall. So I do it with the help of enzymes. To break a bacterial cell, even bacterial cells have cell wall, all monidins have cell wall except your mycoplasma, okay. So bacterial cell, I break it open with the help of lysozyme, and fungal cell with the help of chitinase, and plant cell with the help of cellulase. So anything that ends in ASC is an enzyme, and lysozyme, so this is also an enzyme. Yes, 𝐷𝐺𝐸, bye bye, take care. Right? So now the cell, so you have broken the cell wall. Now you have to break the other content. So you have to destroy the proteins and all, because you need only the genetic material. So you treat the cell with protease and ribonuclease so that you destroy all proteins and RNA, and at the end you need only the DNA. So now only the DNA remains, and the DNA is pretty concentrated, so it is very, very viscous. So immediately you add chilled ethanol so that it precipitates, and it is in the form of semi-viscous form. Okay, so this is how you isolate DNA. So the first step in the process of recombinant DNA technology . So you have to isolate the DNA or the plasmid, right? So this is my cell. So cells have cell wall in most cases, right? And this is my nucleus. So inside my nucleus is where I have my DNA. So I have a cell wall and I have the plasma membrane. So first I have to break open the cell wall. Depending on which organism it is, accordingly I will use the enzymes correspondingly. Yes, okay. Isolation is to take, isolate is to separate, to remove, to extract. That is what isolation means. Okay, so this is Vishal. So finally I found out that is Vishal. Alright Vishal, so thank you so much for joining us. Okay, so if it's a plant cell, I use cellulase enzyme. If it's a bacterial cell, I use lysozyme. If it's a fungal cell, I use chitinase. Okay, so now that I have broken the cell wall, now I have to remove all the proteins and the RNA that is present inside a cell, right? So how do I remove all the proteins and the RNA? The nuclear membrane is also a protein, right? So I treat with an enzyme that is called as protease, and I treat it with RNase. So all my proteins and my RNA are dead. So now the remaining is my DNA, and this DNA is concentrated. So when I add chilled ethanol, so now this precipitates. So this is my purified DNA. So to this DNA, I will give my restriction enzyme. Alright? Understood? Fine. So we go to the next step, that is amplification of this gene of interest using PCR. So PCR is polymerase chain reaction, that is the full form. So Kary-Mullis is a scientist who turned this idea into reality, and he was awarded the Nobel Prize for the same. So PCR actually it enables the production or amplification of billions of copies of an original piece of DNA in a tube within minutes or hours. So this is a PCR machine. So PCR is pretty common because in the last lockdown also, so we had to take a COVID test, that is your RT-PCR. So RT-PCR is reverse transcriptase PCR or real-time PCR, but PCR stands for polymerase chain reaction. So basically just to see if your viral DNA has integrated along with your host DNA. So that is why we amplify that region where we suspect it, and that is your RT-PCR. Alright, so how is PCR happening? So here we go. So I have a figure. Yeah, so this is how PCR happens. So this is my entire DNA. We have isolated DNA. Yes, so we have just isolated DNA. So from the isolated DNA, I want only this region amplified. So I want to produce many copies of the segment that is red in color. So how do I do it? So PCR is done in 3 methods. The first method is denaturation. So first these 2 strands will be separated. So DNA is double-stranded, both the strands are separated. That is your denaturation. So I give very high temperature. A temperature as high as 72 degrees I give. Okay, so at 72 degrees the 2 strands separates from each other or they denature, right? So denaturing is done. So now I give something called as a primer. So primer is a short sequence that you have seen in your DNA replication process. So we have done molecular bases, so you know how DNA replicates. So there also we use a primer. So primer is a short sequence which has sequences complementary to your template strand. So this is your template, right? So you have primer. So primer has sequences complementary to your template strand. So now this is your template. Yes, W, you're right. Those are the 3 steps of PCR and this is your template. So you have a small primer. So we go and stick here, right? So this is annealing. So in annealing, the temperature comes down to 56 degrees. So now your DNA polymerase, because in our normal regulation process, so in our normal replication process, we have DNA polymerase which comes and extends this line which polymerizes the strand, right? So now here we have to do it outside the body, so we cannot take our DNA polymerase. We use tag polymerase. So tag polymerase comes from a bacteria that is called as Thermus aquaticus. So it is an archaebacteria which stays in hot springs, alright? Yes? Fine. So now this tag polymerase polymerizes the strand. So we also know DNA polymerase only takes the nucleotide and adds, it does not synthesize the nucleotide. So I have to give the nucleotide artificially so that the tag polymerase adds, and I have 2 copies of this version. So this cycle repeats 2 power n times and we normally set the cycle to 30 or 15. So if I have 30 cycles, so it will give me so many copies of it, right? So there are 3 steps of PCR. The first step is denaturation. I separate the 2 strands. The second step is annealing. So where the primer comes and binds. The third step is my polymerization step or my extension step where my tag polymerase extends the DNTP. So now what are the requirements of my PCR? So first I need the template strand which I need to amplify. The template strand or the gene of interest that I need to amplify. So I need this portion first, right? So only then I know which one to amplify. So second one is I need primers based on my gene of interest. Number 3, I need tag polymerases. And number 4, I need my DATP or DGTP, DCTP or DTP. All these are nucleotides. The nucleotides should be added because the tag polymerase just picks one nucleotide and adds it in the chain. So these are my requirements, and at the end I have many copies of my PCR. So I have a template strand. So first step is denaturation. The 2 strands separate, right? So once the 2 strands separates, what happens? The primer comes and binds. So this is my tag polymerase, but the tag polymerase waits. The tag polymerase does not function here. So quickly the primers come and bind. So once the primers come and bind, again the temperature goes to extension step. Extension step, the temperature is pretty high. So in denaturation, it is more than 72 degrees. We take it as much as to 95 degrees. So that is the temperature in denaturation. Alright? Yeah. So this is how it will categorize the polymerization step. Alright. So this is your PCR. So now the last before step, culturing of the host cells in the nutrient medium. So how does this happen? So the ultimate aim is to produce desirable proteins in a large scale, hence the need for recombinant DNA to be expressed. So there is no point in doing all this if the recombinant DNA does not give us the desired product. So if you are inserting a gene from Einstein in my body, that gene has to be expressed. So I have to be as good in physics as Einstein is, right? But if that does not happen, if the gene is not expressed, the entire process is waste, right? Yes. So hence the need for recombinant DNA has to be expressed. So that is the ultimate step, right? So though you go to coaching, though you spend so much of money and time, at the end if you don't get result, that's what everyone will look into it. So here also the output is needed. Wow, thank you W. So I'm really happy that you're enjoying my classes. Okay. So if any protein encoding gene is expressed in a heterozygous host, that is called as a recombinant protein. Suppose if I want to produce a human protein, say for example insulin, so insulin is a peptide hormone. So if I put the insulin gene inside a bacteria and if that is getting expressed inside the bacteria, that is my recombinant protein because I have inserted into someone else, right? Yes. So cells are multiplied in continuous culture system. They remain active in log exponential phase and they produce larger biomass. Suppose say I have put the recombinant plasmid inside bacteria, right? And that recombinant plasmid has a gene that codes for my insulin. So the bacteria will express the insulin, for which I have to grow the bacteria in large numbers. So these vessels are called as fermenters. So food is given to grow the bacteria. So the bacteria initially takes time to get acclimatized to the situation and then it quickly begins to grow exponentially. So it produces maximum biomass. That is, it divides and produces and multiplies more of its time, right? So to produce large volumes, bioreactors or fermenters are used. Fine. So vessels in which raw materials are biologically converted into specific products, individual enzymes, microbial plant, animal or human cell. So bioreactors provide the optimal growth conditions for the desired product. Suppose I am trying to cultivate bacteria in a fermenter, there should not be any fungal growth because for example penicillium inhibits bacterial growth, right? So I should make sure I have sterile environment. That is my chemical engineering, right? So in the start we read who gave birth to biotechnology, right? So commonly used bioreactor is STID type. So in big companies you have as big bioreactor as this. So these are the two bioreactors that is present. This is STID tank. So constantly the contents are stirred so that there is no clumps. So if you have seen your mom making halwa, she continuously keeps stirring it so that the halwa does not clot and it does not form clumps, right? So similarly here we continuously keep agitating it to prevent any clots, right? So this is my simple STID tank and this is parched STID tank reactor. So I pass gas so that the contents keep moving. Alright. So the last step here is downstream processing. Suppose if my bacteria has produced the enzyme insulin. Okay, so insulin is here. But ultimately in the market, I won't sell insulin along with bacteria, right? So I need isolated insulin. So I'll have to purify the insulin and kill the bacteria here. Okay, inside the bacteria insulin is produced because inside the bacteria only I have my recombinant plasmid. So this expresses and this produces insulin inside the bacteria. Now my duty is to isolate only the insulin because commercially I have to sell only insulin. So this is called as downstream processing. So after biosynthetic stage is completed, the process is ready for marketing. From before marketing, you should do separation and purification step. Alright. So this is where your biotechnology ends. So we have successfully completed biotechnology, and we have seen it in a stepwise way. We have seen it how each process happens separately. How do you cross check? How do you go about each and every step? So this is much easier to be said than to be done, because it is a lot cost effective, cost expensive, and it takes time. It takes a lot to learn, or it takes a lot to become skilled in this technique, because in one go you will not be able to handle everything. Alright, so it's a very beautiful chapter. So in the upcoming chapter, that is biotechnology and its application, we are going to see how this idea is used practically in everyday life. Yes, biotechnology has been useful in solving practical problems. Let's see how. That is for Wednesday. Alright. Mentee. Okay, so we'll have a mentee on Friday. Alright. So Friday 4:15 we'll have a mentee. Alright. So thank you so much, and if you genuinely liked my session and if you genuinely liked my way of teaching, so please don't forget to like, share and subscribe. I will meet you on Wednesday at 4:15. So until then, this is me wishing you to be safe, stay healthy and stay happy. So thank you once again. I will meet you on Wednesday. Yes, W, bye and thank you guys.